Good afternoon, my name is Pablo Díaz. I work as a plankton ecologist for marine skull and science, and today I would like to talk about the work we're doing on ocean acidification and calcifying plankton in Scottish coastal waters. Before that, I would like to give you some background about Marine Scotland. Marine Scotland is the directorate, the Scottish government responsible for the integrated management of Scotland seas. I work within the scientific division of Marine Scotland, based at the Marine Laboratory in Aberdeen. Ocean acidification and the associated alteration to seawater carbonate chemistry poses a significant threat for many marine organisms, implying broader implications for ocean ecosystems through, for example, food webs and habitats. The potential impacts of ocean acidification on plankton include a number of different processes, but probably one of the most significant ones is on the formation of shells and skeletons by plankton calcifiers, either causing its dissolution or making more or increasing the energetic cost to build them. The Scottish Coastal Observatory is operated by Marine Scotland Science and has been monitoring environmental parameters in Scotland's coastal waters since 1997. A combination of temperature, salinity, nutrients, pigments, phytoplankton and zooplankton communities are measured at selected sites around the Scottish coast. The data collected at the Scottish Coastal Observatory play an important role in meeting the Scottish Government requirements for a number of statutory monitoring and reporting obligations about the status of the marine environment. Our study about the potential impacts of ocean acidification has focused on the Scottish Coastal Observatory monitoring site at Stonehaven. Located in the northeast coast of Scotland, physical, chemical and biological data and samples are collected weekly, weather dependent. Additional seawater samples have been collected since 2009 to analyze the carbonate chemistry parameters at the site, including dissolved inorganic carbon, total alkalinity, as well as calcium carbonate saturation. The monthly average variation of pH values during the year at Stonehaven showed a clear seasonal pattern in association with the variation of phytoplankton biomass in the water. In this case, this is described as concentration of chlorophyll A. Lower values of pH are observed during autumn-winter, increasing in early spring and reaching their maximum values in late spring, early summer, coinciding with the initiation or beginning of the phytoplankton bloom, and decrease from late summer till autumn and winter. The calcite and aragonite concentration at Stonehaven show a very similar seasonal pattern, with the higher values observed in spring and be lower in winter. It is worth noting that these values are about the threshold 1 during the whole year, indicating that the seawater at Stonehaven is supersaturated with respect to calcium carbonate. Considering the general assumption that calcification and calcium carbonate in the seawater are positively correlated, it is reasonable to assume that during the studied period, the carbonate conditions at Stonehaven were not a limiting factor for calcifying organisms at the site. However, the carbonate chemistry parameters at Stonehaven, as it occurs in other coastal systems, show a strong variability at different timescales, including interannual, monthly, and weekly. On the other hand, although consistent long-term series are required to calculate robust accurate trends, the dataset today reveals a pH decreasing tendency with marked short-term fluctuations at the site. To assess the potential impacts of this variability on plankton at Stonehaven, we have focused on the main calcifying groups present at the site by analyzing the morphology of Coccolithophores and the shell integrity of Pelagic gastropod and bivalve larvae, and investigating the relationship between carbonate chemistry parameters at the site. I should mention that the pelagic gastropod community include the pteropods, which are animals that spend the whole life in the plankton, as well as the planktonic larvae of otherwise gastropod benthic species. For that, specimens isolated from archive samples collected monthly from Stonehaven between 2010 and 2013 were analyzed using scanning electron microscopy. The analysis of the Coccolithophore community at Stonehaven showed the presence of several species, with the Millania oxley being the most abundant one. The SEM Pictures also showed the presence of 3 different morphotypes of Emiliania axillary attending at the level of calcification, with the slightly calcified type B, the more heavily calcified type A, and an over-calcified form of type A. The monthly average distribution of the abundance of these morphotypes for the study period showed a clear seasonal pattern, with a transition in dominance from less calcified form, type B, to more heavily calcified forms between spring and winter. Although we did not observe any significant relationship between carbonate chemistry parameters and the abundance of Emiliania axillary morphotypes, the comparison of the morphotype seasonality with ocean acidification parameters showed the predominance of less calcified forms under higher calcite concentrations, as well as over-calcified types under lower calcite concentration. Furthermore, when the seasonality of Emiliania axillary morphotypes is compared with the variation of pH at the site, it's possible to observe that the most heavily calcifying forms predominated in winter under lower pH conditions. These results contradict the general theory that Coccolithophore calcification is positively correlated to the calcium carbonate concentration. In any case, it's worth to remember that the conditions at the site during the study period showed supersaturation with respect to calcite in the seawater. The shell integrity of the pelagic gastropods and bivalve larvae was assessed by looking at the shell structure and the SEM. If any evidence of dissolution was observed, the shells were categorized into 2 levels of shell damage, with mild and severe damage corresponding to corrosion affecting to the outer and inner layers respectively. The SEM pictures of the pelagic gastropods community reveal the presence of adult specimens of 2 theropod species, Limacina elicina and Limacina retroversa, as well as the presence of theropod larvae and the larvae of other species of benthic pelagic gastropods. The SEM analysis revealed evidence of shell dissolution in all analyzed species. Although the proportion of severe damage was similar in gastropod larvae and theropods, there were significant differences within those groups. For example, in the case of the pelagic larvae of otherwise benthic species, severe damage was observed in most of the specimens of GOA-ON that we analyzed, while no dissolution was observed in Rhizoa. Our analysis also showed significantly higher proportions of shell damage in theropod larvae compared to adult specimens. Therefore, our results supported the previous studies indicating that the response to ocean acidification may vary even within closely related species, and that early life stages are more sensitive to the potential impacts of ocean acidification. The monthly distribution of the proportion of shell dissolution showed evidence of shell damage during the whole study period, despite the seawater being supersaturated with respect to aragonite. Although we did not observe any relationship with pH, most of the severe damage was observed during winter, coinciding with decreasing periods of carbonate concentration, as well as the sporadic decreases as observed, for example, in summer 2012. These results suggest that seasonal and short-term changes in carbonate chemistry may affect the shell integrity of plankton calcifiers, even under higher saturation values than previously assumed. The SEM analysis of the bivalve larvae community at Stonehaven also showed the presence of several species, including some of commercial interest for aquaculture and fishery industries, such as mussels, oysters, and scallops. However, the identification of the bivalve larvae required the observation of the valves and the hinges. This required the separation of the valves using several chemicals, which may result in the damages on the shell integrity. So unfortunately, in this case, it wasn't possible to carry out the analysis of the same specimens, either for the taxonomic identification as well as for the shell integrity. The analysis of the bivalve larvae shell integrity supported the results observed regarding the pelagic gastropods. with again evidence of severe shell corrosion observed during the whole studied period. As it happened in the case of the pelagic gastropods, the severe damage was observed mostly associated with seasonal decreases in the aragonite concentration, as well as associated with sporadic decreases of aragonite, as it happened in summer of 2012 and in 2013. The apparent contradiction observed in our study between chemical and biological saturation thresholds for aragonite highlights the complexity of defining definitive thresholds for biogenic calcification, supporting the idea of species-specific rather than absolute carbonate thresholds for biotic calcification. The comparison of our studies carried out on pelagic gastropods and bivalve larvae at Stonehaven shows significant differences in the proportion of shell dissolution between these two groups, with higher proportion of severe damage observed in the bivalve shells that reached almost 50% of the specimens analyzed, while in the pelagic gastropods it was approximately of 33%. These results suggest that bivalve larvae are more sensitive to changes in the carbonate chemistry than pelagic gastropods. Model projections in the novel NotC predict occasional near undersaturation and seasonal undersaturation conditions by mid and late century in Stonehaven. These projected scenarios and the vulnerability of bivalve early life stages raise concern of the potential carryover consequences for bivalve populations in the North Sea and flag potential consequences for the shellfish aquaculture industry. This work highlights the value of sustained observations to distinguish ocean acidification changes from natural variability, as well as to assess the potential impacts of ocean acidification on marine ecosystems. In any case, further analysis of time series data will be necessary to assess potential implications for the ecosystem and for the shellfish fisheries industries in the region. To address these questions, Marine School and Science continue with the monitoring on carbonate chemistry parameters and calcifying plankton at Stonehaven, and has recently expanded its ocean acidification monitoring program at the Scottish Coastal Observatory monitoring site at Lough Kew in the East Coast, with the mooring of a CO2 sensor. Further information on this research can be found at the Scottish Coastal Observatory website, as well as in the following papers. Thank you.